Okay, so we're going to streak out some cells, and this is done to isolate individual cells. It's kind of redundant, I guess. We're assuming that these colonies started from single cells, and then those single cells would, of course, be genetically distinct from their neighbors. But it's good practice to do every once in a while just to make sure you get a genetically uniform version. So if any mutations are present in the colony, you can isolate another colony that results from a single cell, and the colony grows up as a result of mitosis, right? So those would be, as long as cell division is working properly and cell replication, those colonies would be genetically uniform across. So we're going to do that, and then we'll have colonies growing up on these plates. So these are the target colonies I'm going to pick from, and these are a couple of plates that I've made with 4 quadrants in them to streak the cells onto. One of them is labeled wild type. Sorry, that's upside down, WT. This is from the unmutated strain that's never been exposed to ultraviolet light. And then A, B, C, D, E, F, G are 7 strains that we suspect of being formamide sensitive that also were exposed to ultraviolet light. So sometimes your techniques are not too fancy. The kind of standard technique for this is a toothpick. So we're going to use these toothpicks that have been sterilized. This is the lid that I took off. So the lid has autoclave tape that tells me it's been sterilized. So even though they're made out of wood and probably had their share of bacteria on them back in the day, they've been sterilized, so there shouldn't be anything on there now. And I've got them on. You can even see the kind of the remnants of the area that I cleaned with alcohol here. So it should be pretty clean. We're going to try to grab these just barely, so the one that's easiest to grab. The tip is going to come into contact with my hand, but the stuff that's down inside of the beaker here should stay pretty sterile. All right, let's do our control first. To grab a colony, you want to get one that's genetically distinct from its neighbors. So I'm going to grab this one here. You really don't need a whole lot. Actually, the target at the end of this is to get a really small number of cells. We're going to make it so that we dilute these out kind of, and that at the end of this, we're spreading individual cells out onto the agar. So this first streak is going to be a goopy streak on the wild type. Just across there, you can kind of see where that's been. Really, with all this, you don't have to see it. You got to kind of believe where you've been. So you're not going to see the cells. You're not going to see any evidence that you've transferred any material, but you're going to see it next time after this grows up. So that just one streak puts the cells on there, and we're going to do a couple different streaks following that. So I'm going to use 2 sides of this toothpick, and each side is now free of cells. So next side, I'm just going to do a line straight down, and I'm going to flip it over and do a series of ziggies, basically filling that triangular space by passing over that line that I drew. So by smearing those cells out, I've separated them from each other, and hopefully somewhere in here, one single cell will grow up into a colony. All right, so that's our, that was our control. Now we're going to take our suspected mutants here. From these plates, I've got them identified on the back, so we're going to take A from here. So same thing, pick now to the A quadrant. And I'm going to flip this over and use the back side. Now I'm going to need a new toothpick. And you want to know where you've been, kind of make a consistent shape on these, and avoid doing what I just did, which is cutting into the agar. Sometimes a little bit of that's maybe inevitable. All right, so that was A, the A colony, coming over here to B. So I've taken it, you can see the place where I've taken it from. The colony gets a little deformed. All right, so we're not going to see the cells today, but after we grow them up, They should come out pretty densely on here. Densely at the top. And then if we've done this process correctly, much less dense as we move on to the rest of the plate. So you can kind of see how I'm handling this. I've got it auger side up, and I can just pick up the auger side as I need to and expose that surface. Allows me to do this with two hands pretty efficiently. Our next suspected mutant is on a different plate, so I'm going to go over here to mutant number C. Grab it from this colony there. And streak. All right, down. And then ziggy, 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 ziggy. Okay, so we got 1, 2, 3, 4 quadrants streaked on there. Gonna put that in the incubator and grow that up. I'll do another plate, but it's all the same stuff from here on out, so I'll save you the trouble of having to watch that.